Hello dear students, let's take today's topic. Today's topic is physiochemical events during fertilization. Already we took this topic and we discussed about sperm lysis. These are the enzymes, a collective name of the enzymes which are released by the sperm to fulfill the process of fertilization, to enabling them so that they can do fertilization, they can meet female gametes. Now let's take some other events which are happening at the time of the fertilization. See that word physiochemical events. So physio means we are talking about various kinds of movements, and chemical means we are talking about various kinds of enzyme. We know that fertilization is a dynamic process. Dynamic means which are that one movement. We are talking about movement, dynamicity. It means we have a various kinds of movements at the time of the fertilization. So that is why we will understand about various kinds of movements, and then various enzymes are also secreted to control the process. And then we have a chemical. So physiochemical events we are discussing. Last video I taught you about sperm lysis. Let's take the various changes which are happening at the time of the fertilization. So we'll take one by one. Let's revise that one. The structure of female gametes we'll discuss already. So we'll take one by one. See that one. And inside we have oocyte, secondary oocyte. This is zona pellucida, and this one is known as vitelline membrane. And then we have this one is known as plasma membrane, and we can also call egg membrane. And in between egg membrane and vitelline membrane, you can see there is a space here. This is the space here. You can see this is the space there. And this space is known as perivitelline space. Perivitelline space. This is the structure I already taught you, and that here outside we have corona radiator. So leave that corona radiator. When we have the striking of the sperm, the next step what we are getting, see that one, the same structure. When we have the striking of the sperm, we have the various changes in the case of that female gamete, that egg. We will have that first one, depolarization, and that depolarization enabling sperm to enter. So that depolarization will happen. So after depolarization, what is happening? You can understand this diagram. Focus on this diagram. Here that we have egg membrane, and that below egg membrane there are various cortical granules are there. Various cortical granules. So these cortical granules, they start releasing their secretion, and when they release their secretion into that perivitelline space here, that perivitelline space. So what they are doing, they are making vitelline membrane as a fertilization membrane. So try to focus here. This vitelline membrane is becoming fertilization membrane. What we are calling fertilization membrane. And that sperm can enter inside. After becoming that vitelline membrane into fertilization membrane, and how this change is happening due to that release of the chemicals which are present in the cortical granules. And where the situation of the cortical granules? That below the egg membrane there are granules. So perivitelline membrane, now it is known as fertilization membrane. So what will happen after that? We have the entry of sperm, and that when we have the entry of sperm, perivitelline membrane becomes harder, and it is responsible to prevent other sperms, because we have 62 million sperms are released at the time of the mating, at the time of the ejaculation. When that ejaculation is the term, that release of the sperm. So when we have a release of the sperm, a normal count is that in millions, 62 millions. So vitelline membrane is becoming harder, and this hardening after the entry of sperm, when we see the next diagram, what is happening, try to get and understand. This one is, so when sperm enter this vitelline membrane, that is fertilization membrane, it is becoming harder to prevent the entry of other sperms. So there are other sperms, after the entry of that one sperm, other sperms cannot enter inside. You can see there are various, so many sperms are surrounding that vitelline membrane, but due to that change, after the entry of one sperm, it becomes too hard and these sperms cannot enter. So what is the concept I am trying to explain you? To avoid polyspermy. What is polyspermy? Entry of many sperms. It is avoided after the hardening of the fertilization membrane. So that is the concept. I hope you have understood. So this is the relevance of this hardening of that one. Okay, then after we have that activation of ovum. Try to get that one. I taught you about oogenesis. We had 2 times of meiosis, and at that time we had the formation of polar bodies. So how many polar bodies are formed? 3 polar bodies are formed, and first meiotic division followed by second meiotic division. So here is a bit contradiction. That is known as secondary oocyte. Inside this is secondary oocyte, and it starts that one second meiotic division. So whenever we have second meiotic division, it will form that one functional female gamete. Try to get this one. We did not have formation of that functional female gamete at the time of the oogenesis. We had a formation of the secondary oocyte, and at the 14th day of the menstrual cycle, this secondary oocyte was released and situated at the fallopian tube. Female body, I taught you, it is designed in a way so that it can bear also development of embryo, so that all changes are happening. So we will have the activation of the ovum. Here you can see, after the entry of the sperm, this sperm activates secondary oocytes. And which kind of activation we have? Then it starts dividing, that is meiotic second division will take place. So whenever we have the meiotic second division, try to get this one. Whenever we have the meiotic second division, so we have the formation of one functional female nucleus, and there is one polar body. So polar body is moved out, and we have the entry of that sperm nucleus, and then we have the degeneration of the tail portion. There is no need to tail portion, so it degenerates. So polar body is formed. At that point we call animal pole, where we have the formation of the polar body. Try to understand, where we have the formation of the polar body, we call animal pole. It is very important to understand. Polar body is formed after the activation of the ovum. That's why whenever we have the entry of the sperm, then sperm is activated ovum to do, because we will not have the female nucleus, we will have only secondary oocyte. So after that activation, second meiotic division take place, and second meiotic division will result into the formation of the polar body and one female nucleus. So that polar body, where it is situated, it is known as animal pole. So are you getting my point? This tail is degenerates. So this one is known as female pronucleus. What we call female pronucleus. And this one is known as male pronucleus. Male pronucleus. And these pronucleus, they start combining together, and then we'll have the formation of the zygote. So that is the concept, my dear students. Animal pole have you got? So opposite animal pole, we have a vegetal pole. You need to only understand. You need to understand what does it mean, animal pole. Animal pole is the pole where we have the formation of the polar body. How polar body is formed? After the second meiotic division. We did not have second meiotic division at the time of the oogenesis. We have the second meiotic division whenever sperms reach into fallopian tube. So it activates secondary oocytes, and after the activation of the secondary oocytes, will have a second meiotic division. Suppose when sperm is not reaching, that secondary oocyte is also coming into the uterus, and we know that we have the endometrium lining. Menstrual cycle, I taught you, endometrium lining was that became thick. So we have the slashing of the endometrium lining, slashing of the blood vessels, and along with endometrium lining and that blood, this secondary oocyte will also come out. So we will not have the release of the functional female gamete.